Hello, everyone. I would like, as a start, I would like to thank Professor Feza Korkusuts for inviting me to give this talk. Thank you, Feza. You made me really study a lot for this. I'm an orthopedic surgeon with a special interest in osteoarthritis, and therefore my knowledge and the practical terms of this study are limited, and I excuse myself for the lack of technicalities that I can provide during the talk. I speak on behalf of the brilliant scholars who performed the experimental part of the project, such as Seren Özkul Kokac and Yakut Akyon Yilmaz, to name a few. I am personally grateful to their work and expertise, and the paper resulting from the study is today submitted for publication. This is a collaborative study with Hasseltreffe University and Rotte Pharma Biotech company in Monza. The idea of the study came to me a few years ago while reading a paper on the presence of bacterial DNA in the intervertebral disc. I thought a low oxygen, highly structured tissue, bacterial DNA, why? And then the further question was then, is the literature reporting the presence of bacterial DNA in joints, in normal joints, in non-aseptic joints? Today I will discuss the emerging role of the microbiota in osteoarthritis, and I will briefly review current concepts on osteoarthritis, and then I will introduce the gut joint axis and then present the original data from our synovial fluid microbiome studies. Just for the sake of clarification, the term microbiome describes the entire habitat, including the microorganisms that are inside the organ and their genomes and so forth. While the microbiota refers to the viable microorganisms that comprise these communities, and the dysbiosis is the imbalance of the microbiota. The osteoarthritis fills up general practitioner clinic and orthopedic non-trauma clinic, and the knee in particular recapitulates the pathology well. Osteoarthritis is highly prevalent and has a major socioeconomic impact in terms of loss of work hours, total money spent for the cure from patients' healthcare budget, and so forth. Increasing evidence shows that inflammatory and systemic factors contribute to disease onset and progression. And this on the left is a crippling late stage of knee osteoarthritis shown on standard X-ray. Osteoarthritis is no longer considered a purely mechanical disease. It should be viewed as a multifactorial condition rather than a single cause disease. Mechanical stress, metabolic alterations, inflammation, gender, genetics, and aging interact to contribute differently across patients. This is a cartoon from an old Francis Berenbaum paper, which is still very actual, really. This is the whole shaban, the whole circus that happens in the joint, in the articular space of the joint, and it shows the involvement of the synovium in osteoarthritis pathophysiology. Not only synovium, but also synovial fluid, I should say. Products of cartilage breakdown that are released into the synovial fluid are phagocyzed by synovial cells, and the synovial cells amplify inflammation. They activate pro-inflammatory mediators and in the production of proteolytic enzymes, which in turn are aggressive towards cartilage. The synovium produces also anti-inflammatory cytokines, and these also have effect on the overall balance of inflammation and breakdown. The synovium contributes to the formation of osteophytes, as you can see, through bone morphogenic proteins. Because different pathogenic mechanisms can lead to similar osteoarthritic phenotype, osteoarthritis remains a therapeutic challenge. To date, there is no disease-modifying treatment that has been proven effective, and current therapies remain largely symptomatic. To address the serious condition whose public health impact, as I said, is expected to increase with time, increase with the mean age, and increase due to the higher demand of older people. There's an urgent need for innovative approaches, and these approaches should go also through a thinking out of the box for this pathology, not just being concentrated on the joint itself. And another very important thing is that the relevance of the study should be focused on the first steps of osteoarthritis. And this is because cartilage cells, when they die and when the cartilage dies, we don't have many options to reconstruct, and also reconstruction is not exactly efficient as it was the normal cartilage. It's a little bit like we study dementia, and we want to stop the disease before the patient becomes really demented, because the parallel holds, because you can put more neurons in a brain, but this is not the neurons that are going to be able to live in an efficient situation for the patient. The same thing is for chondrocytes so far. And this figure was recently published by an ORC group paper, and it demonstrates the progression of osteoarthritis, and the focus is on the beginning of the progression of the disease, that should be then the part of the disease where we want to concentrate in order to have more efficient, effective treatments, the yellow column. Let's shift gears now and talk a bit about the human microbiota. The gut microbiota influences the host immune homeostasis. This biosis, again, has been implicated in metabolic, cardiovascular, and inflammatory diseases, raising interest also in musculoskeletal conditions. The number of microbial cells that are in an individual has been calculated to be more or less like the number of eukaryotic cells, so there's quite a bunch. But the total weight of the microbiota should be approximately 0.2 kilograms. In either case, the microbiota represents a significant source of non-host biological material. And the microbiota undertakes essential biological processes, and thus it is unsurprising that a number of disease states are associated with changes in microbiome composition. This is just a cartoon to say that the gut microbiota and the metabolic and immunologic state all together and influencing each other play a role in the promotion of several chronic disease states such as metabolic disorders, cancer, inflammatory articular disease, and cardiovascular disease. There is something still that is above and beyond our knowledge regarding the bacterial role in the well-being of the individual, and only now we are really starting to envision this. There's a therapeutic relevance on the effect of microbiota modification. And there's plenty of studies regarding that, involving probiotics, exploring conditions such as Parkinson's disease, COVID, atopic dermatitis, and so forth. There are many clinical trials exploring Clostridium difficile infection also. That Clostridium difficile is a gut infection that is very difficult to eradicate, and the idea of using the probiotics is to perturbate the environment for the Clostridium in order to make life difficult for the Clostridium. These methods might be useful for the treatment of a range of diseases in the future, and so we'll see what all those studies will bring us. One example is the inflammatory bowel disease, where the inflammatory bowel disease is normally treated with therapies attempting to target cytokines of the inflammatory cascade or target systemic immunomodulation. And now there's two innovative approaches. One is the fecal microbiota transplantation, a total transplantation of the microbiota that can give remission to the disease, and also the use of recombinant bacteria that might produce factors that might interact with the inflammatory state and downplay the inflammatory state. Cancer is also a relevant part of the microbiota studies. There are attenuated bacterial strains that can function as vaccine. Studies of Salmonella, whose some particular strains of Salmonella can penetrate and grow within tumors. Tumor tissue, and the specific bacterial strains that are engineered to target the stimulator of interferon genes agonist pathway to trigger anti-tumor immunity. There's relevance also regarding metabolic diseases where bioengineered bacteria can bring the missing enzyme and overcome a genetic problem, but in the case, for example, of phenylketonuria and hyperoxaluria. But there are cases where the metabolic problem is not a genetic problem, but as well can be treated as for the case of hyperammonemia that is present in liver failure. And we can have bacteria that take care of the extra ammonium in the guts. In inflammatory arthritis, such as rheumatoid arthritis, the gut microbiota alterations are well established and precede the onset of the joint disease. The complex interactions that might occur between an altered intestinal bacterial flora and an immune system genetically predisposed to autoimmunity and could potentially provide the basis for the development of this systemic inflammatory imbalance that at the end involves the joints. This is rheumatoid arthritis. In the case of osteoarthritis, instead, there's limited evidence, but growing, particularly regarding microbial DNA detected in joint tissue. And it seems like the gut microbiota could be reshaped by some osteoarthritis relevant factors such as aging, gender, diet, obesity. And all this can boost systemic inflammation, suggesting the possibility that the microbiome might be involved in osteoarthritis, while limited convincing studies have validated the possible intervention on gut microbiota. Still, the knowledge of the interaction between the osteoarthritis relevant factors and the gut microbiota, it's another layer of our understanding of the complexity of osteoarthritis pathogenesis, and also provides a new perspective on osteoarthritis investigation. Our study hypothesis is the presence of bacterial DNA that could be detected using sensitive sequencing approaches, whereas fungal signatures would be negligible in osteoarthritis joints. In case we were interested in fungus because there were earlier reports of the presence of funguses, and if we identified fungus DNA, we could have envisioned some sort of protozoan vector for local treatment of joint inflammation. And obviously, protozoa can produce many more complex proteins than bacteria. This study is exploratory, hypothesis generating, and is designated to characterize the microbial DNA. Finally, synovial fluid represents a unique biological compartment that we used in the study, where systemic inflammatory or microbial signals may converge. So we used only patients with synovial effusion. So all the patients that had entered in the study had a synovial effusion at the moment of sampling. The synovial fluid was collected and put at -80 degrees. -80 degrees was critical since at higher temperature, bacterial signature were not retrievable. And this on the left-hand side, in the lower part of the slide, is the first experiment that got us totally excited because we were detecting bacterial DNA in the synovial fluid of a 72-year-old male with grade 3 to 4 osteoarthritis. The DNA PCR band was detected on a 2% agarose gel on a UV box. So bacterial DNA was there. How is that possible? How could this DNA get all the way to the joint? Normally the osteoarthritis lacks on microbial component is some strong point, because the clinicians were unable to observe the growth of microbial colonies of synthetic media, as in septic arthritis. So the above evidence now provides for the existence of bacterial DNA and RNA in classically considered sterile areas, and And so then the bacteria or bacterial nucleic acid may reach the synovial space through the blood. And but the translocation could be a translocation of the whole bacteria that can be further opsonized, leaving the nucleic acid alone, or the nucleic acid might get there by itself without the bacterial carrier, or the microbial translocation can follow a state of dormancy to the unfavorable environmental condition. And then, of course, there's the problem of the sample contamination that I will address later. Using cutting-edge next-generation sequencing and bioinformatic techniques, we found that there was a low biomass environment of bacterial DNA, and then we applied strict negative controls and filtering strategies throughout the analysis in order to avoid the contamination. And at the end, we did not find any fungal DNA, but we found DNA from the common regions of bacteria in 6 out of 11 synovial fluid samples. And this is the studies on the amplicon sequence variant that we applied in order to reduce the risk of contamination. And this is the overview of the cases. There were 9 female, 3 male. Most were grade 2 osteoarthritis, 2 were grade 3, and 1 was grade 4 osteoarthritis. 2 of the patients had diffusion, but not a real osteoarthritis. They just had either meniscal tear or ligament ACL lesion. This is the amplicon sequence variants that shows that there is a microbiome diversity with shared areas among the different grades. There are grades that have a unique taxa and grades that have common taxa. And this also showed the diversity of the bacteria present in the different patients. And here you have the patients, and here you have the diversity of the amplicon sequence variants that are retrieved. In terms of the bacterial composition at the phylum level, there's 3 groups of bacteria present, mostly in Y10, Y11, Y12. These 3 patients, it is Firmicutes that is most expressed. And in patient number 3, the proteobacteria is the most expressed, and in Y5 there is this spirochete, which is present, and in Y6 as well. Then if you go to the genus level, all the bacterial types are shown in the various patients. There's various different DNAs. In the Firmicutes, there's Staphylococcus, Bacillus, Streptococcus, Listeria, Enterococcus, which is prevalent. In the proteobacteria, there's Pseudomonas, Acinetobacter, Salmonella, Morganella. And in the treponema group, there is spirochete, which are highly abundant in patient 5. What of this is environmental? What of this is really synovial? We will address that shortly. Some pattern of distribution of the various bacteria here in the various patients here can be envisioned, and several bacteria generally detected in our synovial osteoarthritis fluid samples are bacteria that have been previously reported in the literature. In contrast, we found genera that were not previously reported and are more likely related to eventual environmental or reagent contaminations. And so then these are all the samples, all the papers that reported the presence of bacteria, and as you can see, there's plenty of bacteria that we reported that were not previously reported in osteoarthritis, but reported eventually in other situations. Such as septic arthritis, on postoperative periprosthetic infection, or so forth. One genus that was new to all in the literature was this Lysinebacillus, which is a totally new entry, and we'll have to figure out why is there and eventually what is it doing. The interpretation of this finding should not be interpreted as a real joint infection, but rather as a microbial DNA signature, which could potentially influence local inflammation. It is well established that the immune system is able to differentiate self from non-self, and it can differentiate self DNA and non-self DNA as well. And so there must be an immune reaction to this DNA, and this could be the base eventually of the chronic inflammation of osteoarthritis, among other things. Our results add a biological plausibility to the gut osteoarthritis axis and suggest new mechanisms linking systemic and joint level inflammation, and opening new research direction in this field. The potential presence of biomarkers that could demonstrate the role for the inflammatory basis of osteoarthritis are important. And of course, they can be targeted in the joint. They can be targeted also on the gut level. And we have to consider that very often these patients end up with an arthroplasty, and the arthroplasty is done possibly on a bacteria environment, and this could be a big issue for the capacity of the bacteria to grow on the prosthetic surfaces. Of course, there's limitations. Limitations mean that results must be interpreted cautiously, and we need larger cohorts, of course. This is often the case. This is always the case. Patients, all the patients had synovial effusion, And this might create a bias because we studied only acutely inflamed joints, and the study is cross-sectional, so it does not have a follow-up on the results. Take-home message. Joints are not necessarily sterile. Even if you cannot grow anything, there might be something there. Bacterial DNA is detectable, not fungal DNA is detectable, and this results support the emerging gut osteoarthritis axis. And I would like to thank all the collaborators and funding institutions. And I would like especially to thank the departments mentioned at the Setup University. I would like to thank my friend Lucio Robati, who sponsored the study with his company Rotte Pharma Biotech. And I would like Beppe Perale for the endless discussion to envision this study in the first place. And thank you all for your attention. It's amazing what you can do with AI these days. Thank you.